Never was an early riser, used to be an up all nighter. Never saw the morning light quite like I do now. Never said no to a party, never started saving money.
Hi everyone, how are you? So today we are going to finish the chapter 2, which is about the chemistry of life, and the final part of the chapter 2, which is about the four major types of macromolecules that are frequently found and that actually constitute the life structurally and functionally. Actually, these the four different types of macromolecules are characteristics that we are going to understand. What's the main purpose that actually, and the reason why I generated a new chapter of this chemistry of life for this semester, kind of impromptu, because especially to the non-science major student, because these macromolecules including carbohydrate, proteins, and lipids, different types of lipids, as well as the DNA and RNA nucleic acids, are the subject of this whole entire semester's course. So you will be encountered with many various different types of such macromolecules. So without clearly understanding each different properties, it will be really difficult to connect their respective functions, and so without knowing them, it would be a disservice to especially the non-science majors. That's the reason why. So don't get intimidated if you haven't had this information in the past. As I emphasized before I begin this chapter, you don't really have to get intimidated by anything of complex looking the chemical details. The big picture is the most important thing, and we are only here to learn a little more about the big guys like proteins, what type of proteins and why those proteins behave like that, and like why there are so many different carbohydrates, and then each of them is utilized in what way, type of thing. But when you do that, if you do not know a little bit about the basis of like chemical bonds and some of the chemistries, then it will be really difficult get things across. Okay, so having said that, let's get them. So these four major classes of macromolecules constitute life. So these macromolecules, how do they are four different types of carbohydrates, lipid, and a weird color coordination of proteins, like having intentionally taking the rainbow colors, pun intended, and a nucleic acid. So these four different types of macromolecules, all of which the one common characteristics are they are as polymers. They all built from the monomers. That's one of the very important the feature that we have to understand first. So all of this starting from monomers, the small things, through the building process. So all these are big polymers. So this arrow, how was it possible to make this arrow go? So here, these are the chemical bond, and the type of reaction is what? Guess what? The anabolic reaction, of course, right? That's the type of relationship that all of these four different types of macromolecules have, right? So once again, as we see this figure, what we can learn about this figure is so many different types of so-called the macromolecules. So for example, like in the case of cell membrane here, if you zoom in the cell membrane, you get to see this actual cell membrane, which is made of a type of lipid. But not only that, specific different types of protein is like inserted. We can usually describe this as an embedded protein. And also further, on top of protein, usually these proteins have some of these carbohydrates attached. So all these macromolecules interact together to have a very specific, useful function, which is vital for the survival and maintenance of this particular cell. By the way, it happens to be a plant cell, so it contains an additional protective layer outside of the cell membrane, which is a cell wall, and the material making up the cell wall is a type of a carbohydrate. Yeah, we will get to that later. In fact, that is the name of this particular carbohydrate constituting plant cell wall is cellulose. But that is not the only usage and type of carbohydrate you will get to see in the cell. Another example is, look at this starch, which is the end product of active photosynthesis. Then this starch is actually another type of carbohydrate, but it's a storage form of our energy source, which is actually immediately after the photosynthesis, the product is made as a kind of in the form of glucose, which is more familiar to us, but they are in the form of starch for storage material, just like that. So not only that, there are so many other different types of carbohydrates around in the cells. And also, speaking of the proteins, you see this membrane-bound protein and some other proteins like enzyme and some other structural protein over here. Its name is microtubules. It's a long tube-like protein which provides a very important in maintaining the proper structure of the protein. And later in other chapters we are going to take a look at more details of this protein too. And also ribosome is a relatively big structure which is specializing in the protein synthesis. It's actually is made up of two different types of the macromolecules. In that, one is a lot of different types of protein, ribosomal protein, and also several different kinds of RNAs are mixed together to make huge, big snowman-like structure, which is once again is extremely essential for anybody, any living cell, because its function is protein generations like that. So, I think it's just enough to give you an impression about the diversity and importance of all these four different types of macromolecules. And once again, the starting point of these whole macromolecules was the individual molecules, which we in a group define as monomers. And so from monomers to polymers is the way they get to be generated. So to glance, to have a bird's eye view of different properties of for these kinds of macromolecules, probably we can compare. Although, yeah, it's me and nobody else has tried this type of a silly thing. I just several years ago when this program on TV was so popular, I just, well, what the heck, I'll try to make a little silly joke about this and this, how how I just made this comparison, because to some sense, these two are straightforward and obvious. Why this gentleman, I can easily compare this gentleman as a representative of nucleic acid in his personality, which is very much like the chemical personality, I mean the chemical property of a nucleic acid, because one word is boring. So just like this, sorry, but you are boring. Just like this guy, nucleic acid, whether it's a DNA or RNA, especially the DNA. RNA is a little bit better, but DNA is extremely boring, huge macromolecules in terms of the structure, of course. And this one is also very easy to have this kind of particular comparison because of proteins. As this gentleman constantly claims to be himself as a special, yes, proteins are very special among these four different types of macromolecules. So that's the reason why proteins take the most charge of acting as a very peculiar and important cellular functions, including the enzymes and so many different types of transporters and receptors, etc. Because proteins are very special. What? In what? Special in the shape, how they look. So once they can become special in their shape. What I mean by that is a structure. Then they can function very special things. So that's the already very important tight link between the function and structure. And so for these two guys, I just I didn't know actually what to say about this, so I kind of I made up randomly. One might actually try to put this carbohydrate, not I mean the carbohydrate, but especially these lipids to this guy, so that that guy might be a better representative of the lipid because of his big belly. But I think a little different because among all these four polymers, actually lipids are the smallest in its states, the smallest polymers, as we will get to see a little bit later on. So that's how I just ended up putting this lipid tag on him. Sorry, I have a strong sympathy for you as a one of a kind myself, but nothing can be done. Anything, nothing we can do about it. I mean, what I mean by that. So, no hard feeling. Carbohydrates. So, well, just like that, carbohydrate, or we can just try to bring any kind of a silly excuse to justify why this guy represents the carbohydrates, but that's not a moot point. Well, I just want to make some distinction in their peculiar properties, especially on proteins and nucleic acid. That's also you guys' missions accomplished. So thank you. So all this, especially to build this polymer, so you have to start somewhere, starting from assembling each between monomers, right? Here is the monomer. But what kind of chemical bond are you going to use? As we have seen in the previous section of this chapter, these are all made by the covalent bonds, because the covalent bonds property, which is chemically extremely stable. So the covalent bonds are very stable, takes high energy to build, so it's more difficult to break. So it is a very strong bond. the definition of strong in terms of the bond strength is always is about the energy. It takes more energy to build, and it's also very easy to, I mean, it's more difficult to break it. So that's that's what we call okay, that is a strong and stable. And comparatively, on the other hand, like hydrogen bond and van der Waals bond, and those very chemically weak bond, and also ionic bonds too, are very easy to separate and break because their bond strengths are weaker. So once again, when I emphasize, whenever you see this type of a solid line between the elements, then you know, oh, that indicate the covalent bond. Okay? No other chemical bonds are represented by using this solid bond. So this is the privilege, only privilege of for the covalent bond. And the molecules forming the covalent bond can be, as you can see over here, be represented in many different ways. Like you can simply write down the alphabetical constituent of that molecules, then we call that as a molecular formula. But then here, by drawing specific individual line between the two elements, we can specifically at which point and which angles these elements are forming covalent bond. In that, then we call such we can have additional information in this type of chemical formula called the structural formula. And so, like, to make a much nicer, fancier looking structural formula, sometimes this each element is replaced with some specific colored ball, and we call that as a ball and stick model. We have already seen this, which colored ball represent in by convention generally. So like always, the black ball represents carbon, and red oxygen, right? And white, so or hydrogen, and this is the blue one is nitrogen, and sulfur is yellow. Okay. By the way, please do not even try, do not even think about memorizing those. That's useless. Okay. That is not the reason at all to showing you. It's just like when you see this type of thing, then what does this red thing represent? Okay. And usually you will be able to figure it out, so you don't really have to memorize everything by force. And then to illustrate the more realistic view of this each element's the electrical orbital territory, then we can expand this ball and stick model into something more realistic, reflecting some of the more modern aspects of this quantum physics. Then space filling model is sometimes used. So that's the reason why you get to see a different version of this chemical description for an identical molecule. So now, among this covalent bond, to build a polymer starting from those available monomers, in terms of speaking of all these four different types of biologically important macromolecule, there is one very consistent, identical mechanism, rules for this chemical building procedures through the covalent bond formation. The name is condensation, or in some other, it's also called a dehydration. Both of them representing some of the extracting of water. Okay, so condensation and dehydration means both of them means the water escapes from the the participants. Okay, so this covalent bond formation to assemble these macromolecules always involves this condensation reaction. As you have seen this in perhaps in your the handout, lecture handout, this is like each block, individual block, represent a monomer over here, and in order to build them by forming each covalent bond in between, all it takes is to connect between these two. How can you do that? Is so in order to initiate this condensation reaction, which is a type of covalent bond, so these two participating monomers must have an available which can be donated, available the one water molecule worthy of any particular element part. So for example, these two together can make up one water molecule. As long as this type of combination can be generated, then you are good to go. So then in between these two, you guys go out, and then instead we can have one solid connection of a covalent bond in between. So as a consequence, water molecule, one water molecule, one OH, which is hydroxyl, and one hydrogen putting together make up this one water molecule will escape. Okay, that's the basis of this condensation reaction. In other view, also you can follow this condensation reaction over here too in this diagram, which is identical. I'm just showing you to brag my ability of the drawing the even it's not something that I drew. So why did I just put this thing up here? Silly me, sorry. So anyway, this is the same condensation reaction is showing over here. Now, what if I don't want? What if I want or have some kind of refund? Here, you have this. Hey, I want to go back. I don't like this type of really rigid environment. I want to be free once again. Then just go there. Then ask whoever in charge, then ask for this refund. Hey, can I? And then that guy is so generous. So sure, of course. So as long as you get the receipt, then we will just give you a refund. Okay. What's the refund? What would be the receipt? The water molecule. You just as long as you just bring the water molecule back, then it can be reversed. Okay. So this, using one water molecule by addition, then this chemical bond formed by condensation now can be broken, and this reverse procedure is called hydrolysis. Think about this, the actual, try to dissect this vocabulary. Hydro, water. Hydrolysis is something that breaking. So using water, you are breaking something. So that's why it is called hydrolysis. Okay. So this hydrolysis reaction will break down the polymers into individual monomers. So this figure shows the opposite of the condensation, which is the hydrolysis reaction. So, like if you add one water molecule, then the bond between here, these two monomer, can be broken like this. So once again, then each individual monomer can go freely as they want. Okay? So all these carbohydrates, lipids, and protein and nucleic acid are the result of those polymers. Okay, are the result of this condensation reaction, are the same covalent bond involving this condensation reaction. And when each big polymeric such macromolecules is about to be broken, then they have to go through the hydrolysis reactions. But in each classes, the name of such condensation reaction is different. because a slight details of this chemical nature is different. That's that is the only reason why each of them maintaining a different, operating the different name system. So let's try to define the carbohydrate first. Yeah, it is a macromolecules, okay, but why? What kind of a molecule would make carbohydrate as a member of? So what's the qualification? Well, if as a molecule, if you have carbon, many hydrogen, and many oxygen, so these three things are something that are must you have. So you do have to have a carbon, and also you do have to have a hydrogen and oxygen in certain very specific ratio is the definition of a carbohydrate. Actually, that is the why there is the reason why these molecules in the first place is called carbohydrates. Carbohydrates. Okay, these two. And because of that, one the chemical property, whether you are going to be friendly with water or kind of really abhorring the water, would be very important determinant as a like internal residence of the life. So we always have to consider try to constantly remind ourselves a certain particular molecule is hydrophilic or hydrophobic. In the case of a carbohydrate, almost all of these carbohydrate would be basically hydrophilic, which means they are going to be a polar molecule. Why do you think most of these carbohydrates are polar hydrophilic, while this carbohydrates, all of these carbohydrate molecules have too much of this oxygen, and oxygen is always very, very greedy to have electrons. So it will create some of these differentials in terms of this charge distribution. And then, as you can see, as you saw actually in the example of the water molecule, whenever you just have this oxygens involved, then you will almost definitely 100% you can expect that molecule would be polar and hydrophilic. And two major, among many other different miscellaneous functions, carbohydrates' main two functions we can summarize as one is the energy source, either in the immediate source of energy or storage form of energy. So in energy, one is like one we can say is immediate, and the other one is storage. Okay, so both of them carbohydrates provide these both functions, and also very important structural support functions is coming from carbohydrates. Some of the carbohydrates, some examples, like here in this figure that you also have in your handout, is three different classes of carbohydrates as shown over here. One is something called a monosaccharide, and the other one is called a disaccharide and a polysaccharide. That's three. Actually, there is one more in between in terms of the complexity between disaccharide and polysaccharide. There is something called an oligosaccharide that we are going to take a look at briefly, at least shortly after this. So some of the representative of examples are shown over here, some of the sugars that we are going to go through very briefly though, each of them. So this among these three different classes of carbohydrate, one, the most simplest sugars. So common ways, a lot of times it's simply called the simple sugars, but the simple sugars professionally is called monosaccharide. Mono means what this mono means. So it has only one unit of such a carbohydrate. So all these monosaccharides actually defines the core of the carbohydrate, because this is the monomers. The monosaccharides are the monomers from which all these different types of polysaccharides and the disaccharide and oligosaccharides are built. Okay, so this is in individual ingredient. So as per this individual ingredient, if you look at and try to kind of have some common rule, yes, luckily you can find a common rule of all different types of monosaccharides can be described as using this formula specific: C standing for carbon, of course, and H, hydrogen, and oxygen, CH2O, which means you have this 1 to 2 to 1 ratio. You always get to probably, yeah, there might be some a little bit of an exception, but almost in every cases with all these different monosaccharides, this carbon, hydrogen, and oxygen appear in 1 to 2 to 1 ratio, and you put this N over here after the parenthesis. What you mean by that is N equals this number of this, actually you can say this N is a number of carbon. So then in terms of glucose, all you gotta do is put 6 over here, then it can be used to represent the glucose, say 6H12O6, right? So in other words, monosaccharides, among many different monosaccharides, like the differences, mainly differences are actually basically the number of carbons. So monosaccharides usually start from their carbon having only 3. So 3 carbon monosaccharide, and also you can go beyond the 6 carbon, and then you can go 7 and 8 and 9, etc. So more common, more familiar to us among many different monosaccharides include these glucose and fructose, which is found in the fruits, sugar, and galactose, and another type of such monosaccharide, and also ribose sugar. You will get to see the ribose sugar when we discuss the structure of DNA over there. So these are such some of the major representative of the monosaccharides. So at this point, probably by just watching this short movie, we can try to
A simple carbohydrate, or sugar, consists of a carbon backbone with oxygen and hydrogen molecules attached. The number of carbons and the positions of the attached atoms give sugar.
All of a sudden, is there no audio? What's going on? There's a mistake in it. Oh shit. Well, sorry about that. So anyway, having this monosaccharide to assemble to have a larger, more complex sugars like different types of polysaccharide, that is the kind of theme of the short movie. And some of the monosaccharide can have like, even though they have same chemical formula, but slightly different in terms of the position, slight position. So then we call them as a structural or stereoisomer. You don't really have to get into this unnecessary complexity, but simply the thing that you want to know is, hey, what's the difference between, for example, if you wondered what's the difference between fructose and glucose and galactose, those three sugars that I just introduced to you, because from when you look at their chemical formula, and you will be confused because all these three chemical formulas are identical, C6, H12O6, but then how they differ, yeah, this is the difference actually. When you look at them as a structural formula, then you understand, oh, I see. In the case of fructose, this position, this carbon having the double covalent bond with oxygen, is buried in between these two different carbons, whereas in case of the glucose, that same unit, the carbon having this double bond formation with an oxygen, is like pushed at the edge. Well, there is a more professional way of naming these two particular positions, but here in this class we don't really even have to worry about. And so the relationship between glucose and galactose is a little bit more complex, and that I don't really have to explain to these the non-science majors about the stereoisomers, but there are certain things like this, different types of isomers, very similar but not exactly identical molecules. We usually call them the isomers. Yeah, that's that much that you should listen. Okay, so let me move on. Another thing is, well, you will actually get to see many in different areas. Sometimes glucose is represented like in this linear chain form, and some other occasions glucose is represented with this type of ring form. Hey, what's the difference? Yeah, they are the same. This same glucose molecule can change its form itself by inducing itself some kind of specific chemical covalent bond formation to close this, to have this ring structure. Is something that naturally occurring in watery environment, the aqueous solution. And actually this one, this ring form is a little bit more favored, something like more than 60 or 65%. They do exist in this form of a ring structure, and another smaller proportion they do exist in the chain form, and they always have this kind of imbalanced equilibrium with this different ratio. So don't get confused just because you saw two different types of structural formula for the same carbon molecule. So in biology, actually, because of these ring forms are more dominant one, so we usually deal with this ring form, the monosaccharide. So the glucose and fructose, galactose, and another type of monosaccharide and mannose are all in this slide represented in this ring form. Okay, but remember, if you ever get to see for any of this monosaccharide, if you saw in the linear chain form, yeah, that is also a correct, valid form of any different type of monosaccharide, although they are something that exists in smaller proportion in nature, in your cell. And here there are, once again, two very important, important as well, two famous 5-carbon sugar, the carbon having 5 carbons, the monosaccharide, more kind of professional looking name for 5 carbon sugars is usually pentose. And here ribose and deoxyribose are shown because these two different 5 carbon sugars, you are going to see them, both of them, when we get to the DNA and RNA, the nucleic acid structure. One is called ribose and the other one is called the deoxyribose. But if you look closely, the only difference between these two are this area where actually all the carbons are arranged and then like distinguished by having their unique numbering system, and they do have some specific rule of numbering carbon. So this is carbon number 1 carbon and number 2 carbon, 3, 4, 5. Okay, we don't really have to go into more of the details, but actually this number should be the number 1 prime because there is but never mind. Okay, I will. So anyway, so that's why you get to see some of the mysterious number besides the carbon, each carbon. Okay. So 1, 2, and it's just for our own convenience sake. So if we have this specific numbering of carbon, then it's easier to communicate. So hey, so it's exactly in this occasion. So to explain the difference between ribose sugar and so-called deoxyribose sugar, or what we can easily is, hey, the difference is like number 2 carbon number 2, the ribose, in the case of ribose, the OH, which is hydroxyl group is attached to the number 2 carbon, whereas in the case of deoxyribose, the oxygen is missing, only hydrogen is attached to the number 2 carbon. So that's the difference. And actually, that is the reason why this guy is called deoxyribose. D means something that is missing. So oxygen is missing here. So then we can infer that, okay, the ribose is the original. Ribose sugar is the original, and then deoxyribose is something that generated afterwards with some kind of a differentiation. So that's what we can expect just by learning these two differences. Okay. So this discussion will continue a little bit more when we get into the nucleic acid section. Yeah. And some occasions actually each specific details of this chemical attached to around the ring form of this structure is like omitted for the simplicity. So these two, both of them represent the glucose, but here it actually real more correct details of glucose, whereas here the simplified form of the same glucose sometimes, because assuming everybody knows what it is, actually at least everybody know what is at each point the carbons reside. So that's something to know. And also carbon should also have some all those maximum chemical covalent bond with anybody else. So unless specified otherwise, anything obvious like the regular those interaction connections will be omitted. Is the convention. Hey, what's going on over here? What did I do? So that's the reason why you see sometimes have this is a very simplified version of the, especially it is popularly utilized when this ring form of the sugars were described. Okay, and obviously this is the ball and stick model of glucose is also there, and space filling model of the same glucose. See, a little bit of more difficult to easily identify. Oh, that's glucose in this case. but this is much easier. Oh, okay, because you know that there is a 6 carbon and oxygens and hydrogens and the others are attached in this particular fashion way, then you know it's glucose. Okay, so that's a short introduction about the carbohydrates. Here, this is about just a little bit more complex form of the sugar called the double sugars, or more professionally is called disaccharide. Di means 2. So it's like all these disaccharides are the one that consisting of 2 monosaccharides put together through that condensation reaction. So sucrose, lactose, and maltose, these 3 are not the only examples of disaccharides, of course, but these 3 are the most popular, well-known examples of disaccharide. Sucrose, everybody know, the table sugar, and lactose also almost everybody know, because this sugar is in the milk. So the mothers and cows and all those mammals' milk contains a lot of those lactose. So that's the lactose is another name of lactose is a milk sugar for that matter, and maltose is another type of a disaccharide which was made only specifically by 2 glucose molecules, etc. Okay. So these are the different types of structures of lactose and maltose and sucrose and all of these. Okay, don't worry anything about anything, but simply okay, between these 2 monosaccharides there is a connection, a covalent bond made by a condensation reaction, which is you can see over that condensation reaction occurring. So to generate such condensation reaction, you will have to extract all together combined form of a water molecule, and as a consequence you establish this covalent link between them. Okay, so that's how you make this sucrose. But with this type of a seemingly simple chemical reaction, sucrose, the only organism on earth that can synthesize the sucrose is plant. No other animals do not make sucrose. Okay. So if all those sugars that we pour onto everybody, all kind of other different food are actually originally they are the one or only made in the sugar canes or any other plants origin. Although in these days actually some factories can make this sucrose by ferment the reaction with the reactors using some of the yeast, engineered yeast, but that is not the natural product of this sugar, sucrose. In this slide, what you see is, upper chemical structure represents the sucrose, of course, here, and the bottom shows another molecule which is also very famous for some other reason. Its name is called the sucralose. Names are very similar, sucralose and sucrose. Okay. But still, if you look at closely, there are certain differences, of course. Yeah. So here in sucralose, you have this chlorine. The chloride, as a chloride, they are attached to some of the part of this. Originally in sucrose, there were this hydroxyl group was there, but they were all replaced with this chloride. So what? the only point I'm making here, trying to, is the sucralose was artificially made, generated some time ago, like 1970 or something, yeah, in the Britain, in some company to as a artificial sweetener. For a long time, there's been a rumor going around that this chemical sucralose as a an insecticide. Yeah, it's been rumor going around. So what? Because that's very surprising. Hey, what are you doing? Something is going on. I see. Sorry about that. Because this sucrose was artificial, as I said, it's an artificial sweetener. And then if you learn that, because you can see this sucralose on many different cafeteria, including Starbucks probably in these days too, as an artificial sweetener. But then if you knew that, okay, actually the original purpose of making this synthetic version of the sucrose, which is called the sucralose, was to kill the bugs, not to soothe us or to satisfy us. And it would be kind of surprising and shock. Why? But actually it turned out to be not true, a kind of false rumor, even though that was even reported in one of the major US broadcasts like ABC some time ago. But the reason why that type of rumor was going around was because of the same idea of developing this artificial sweetener, I guess, because whether it is to be used as an insecticide or sweetener, artificial sweetener, is that unmetabolizable. Sugar, which means you have this sucrose, then inside of our body, the sucrose will immediately broken down into each part, which is a glucose and fructose. Yeah. However, in the case of this sucralose, we cannot do this. We cannot break this sucralose into two individual components. Why? Because we don't have any enzyme. To break down. So like this hydrolysis or condensation, well, if you think about this condensation, because this condensation reaction is what? The anabolic reaction. Is the anabolic reaction is spontaneous or non-spontaneous? It's non-spontaneous. Okay? You have to provide the energy. So then you may actually think, okay, so for that matter, probably we need some enzyme, so that enzyme can bring some of the energy and do this. May understandable. But in this case of the catabolic reaction, the opposite side of this reaction, which is a catabolic reaction, would be a spontaneous reaction. It will happen by itself. So you would think that it will happen naturally. No, it's not. Naturally, it's not going to happen, even if that is a spontaneous reaction in nature, in biology. So almost every bit of this reaction, even if they are the spontaneous reaction, which is catabolic, then you cannot proceed that chemical reaction without the help of an enzyme. But in the case of this sucralose, we have never seen anything like that. So we didn't have any chance, or we didn't have enough time to evolve an enzyme to deal with this. So we don't have any enzyme. So that is the actual point, that this sucralose can be used as an artificial sweetener. It is 500 times sweeter than the sugar, okay? But it will not give you any calorie contribution burden, because it's not going to be broken. So it will simply give you a 500 times a sweeter taste, and then will escape eventually out of our system, basically, with no harm or no damage made on your health of your body. That's at least originally supposed to be acting like that. Although recently there are several reports that actually saying otherwise, claiming otherwise, that actually sucralose can have some of the serious effect on some part of your area, like including the liver or anything like that. But those report has not been confirmed, so still it's a type of like story going on. But anyway, originally at least, the fact of the matter is you don't have any enzyme that can metabolize this type of artificial macromolecules. Okay. And if we somehow stupid enough to develop this sucralose originally as an insecticide, the basic idea behind this would have been actually the same principle. Insect also don't have anything like this, the enzyme to break this thing. So insect will be taking this sucralose instead of sucrose when they try to intercept actually sucrose was the reason why plant only produce this sucrose is one of the very stable form of sugar. So plant use this sucrose as a transport form of, once they are done with the photosynthesis, they generate this glucose, and then they can easily convert this glucose into fructose, and then they combine and then ship these sucrose to the other area where there is no photosynthesis can be done. Their poor brothers and sisters on the same plant, like roots or young leaves, and so that's the purpose of this sucrose, a transport form. And for an insect, this sucrose would be the ideal material to snatch. So insect will pick and then try to steal the sucrose. But if you spray this sucralose on the plant, then insects don't have to, because there is a sucrose, and then they will just eat a lot of sucrose. But then in fact actually those compound they ate is not a sucrose but the sucralose. So they just kept eating and eating and eating, but still they are hungry, and then they starve to death. Was probably must have been the original idea, if that was the case. Okay. However, it turned out not to be the true. Although actually there is at the time of this development, one of the staff member, the newcomer staff member, was told by the head, the person in charge of this development, that okay, why don't you test this sucralose before we actually finalize this as a legitimate product. So then he did. He tasted, because he just misheard test as a taste, and that's how they discovered this effect of this is 500 times sweeter than the actual sucrose was the original, the anecdote of which is actually true. So is sometimes it's not only us, the foreigners, but the native speakers also like have this kind of miscommunication. So they test this and then they taste. In these days, you can easily find this sucralose in the form of brand name of Splenda. Okay. So that is, and there are other different types of such artificial sweetener, including saccharin and some other aspartame is another such artificial sweeteners. But definitely this sucralose is one of the such. And all of these are, I don't know much thing about this aspartame, but at least for saccharin and sucralose, they are, most of them are actually, especially in the sucralose, the advantage of using this as an artificial sweetener is like unmetabolizable the compound, because we don't have any enzyme to digest this thing. So this complex carbohydrate, complex carbs, sometimes they are called. Once again, the more professional name for them is polysaccharides, made as a result of a more large number of the monosaccharides assembled together, have a huge actually. So we have just seen the disaccharide, which is only the two monosaccharides are combined together through condensation reactions. But here, several hundred, even I guess it is not 1,000, I guess, different monosaccharides molecules are all attached together through the same condensation reaction. By the way, actually, the particular type of a condensation reaction used to build such polymer in carbohydrate is called glycosidic linkage or bond. But once again, who cares about the name? Don't worry about the name, remembering. But the examples of such polysaccharides include the starch we have just seen in the early part of this lecture, and another very similar to starch is glycogen and cellulose, and also chitins are the four more famous. But these are not, once again, these four polysaccharides are not the only polysaccharides. There are several more different types of polysaccharides do exist. So these are, just by looking at okay, so here in one photograph of this young man appearing on the beach, was in somewhere is nice. Where is it? It's somewhere in Florida or anything. Having a look like it's having such a good time. Having, what is this? What kind of a fruit is this? Some tropical fruit. I can't even recognize which one, really. But this photograph can show three different types of such polysaccharides all together. So one is cellulose, we can read a structural component of the plant, but as you can see, the same individual unit of monosaccharides are all attached together in one dimensional, but also across each the long chain to have a very kind of layers of different multiple layers of such polymer is one distinct characteristic of cellulose. So it is one of the main structural components of a polysaccharide. On the other hand, starch, which is abundant in my one of my favorite, one, it's not one of, actually it is my favorite food, which is a French fry, is mainly made of this starch, as we all know. A linear form of such assembly of the same, actually, the type of sugar you see in the case of cellulose is the same as you see in starch. Actually, in fact, it turns out to be what? The glucose. It is a monomeric form that used to build this both polymer of cellulose and starch is the same identical glucose. So it's kind of surprising that same glucose molecule can be used to build starch, which is in texture and the properties and or other differences. It can be clearly distinguished from the cellulose. And another thing is starch. There is a reason why one of the many people like me really like a starch, because we can easily digest starch into individual the monomer, the glucose, and then use as a so starch can be used as a food, right? Actually, starch is made in plant, and the purpose of starch in plant was the storage, as I mentioned earlier in this part of the lecture, was to use as a storage form of once you make all those glucose as a result of photosynthesis, you put them together to have a more stable form of the sugar, so that store them, and then later when they need some supply of energy, then they go back to their garage storage and then the chip down, so they can utilize as an individual glucose as an energy source. We animal do the same thing. So if we eat a lot, excessive amount, so that we have a plenty of leftover glucose, then we convert them into a storage form of just a very starch-like form, which is glycogen. We usually put those glycogen stored in our major storage places, which is liver, but also our muscle is another such area where we do have plenty of this glycogen as an energy storage form. And perhaps, as you can easily notice, in the case of starch, you have this all long chain. Why do you have this curly? Well, if you have long chain, then it will naturally have this curly shape to minimize the tension. So this is the most stable structural shape of this long chain. You will see the same similar thing. I shouldn't say the exact identical same thing, but similar thing in the DNA structure. There is a reason why DNA have this staircase-like spiral form, because it's very long. And in the case of cellulose, you have this not only one-dimensional, but across each different branches of chains have additional the chemical interaction. You notice this chemical interaction is not covalent bond, because as you see, they are not written in solid line. So, this is not. It looks like, what do you think? It's like a hydrogen bond, right? And in the case of, that zoomed photograph is to show simply the difference between starch and cellulose, even though they are basically made of the same type of monomer, which is glucose. But to illustrate the slight difference between this starch and the glycogen is simply they are virtually identical compound, except in the case of a glycogen, they do have a little bit more extensive, something like branching is. In starch, especially in the main form of the starch, there are two different forms of the starch. We don't really have to go into that detail. Especially that is mainly, almost exclusively formed in single linear chain form. But in glycogen, it is a little bit different in that they do have a lot of those branching. So that's the only difference. Other than that, the starch and glycogens are like the same thing. Even though animal and plants are like, especially those animals that make this glycogen, and also, I should say, animal is not the only the organism use the glycogen as a storage form of the sugar. Also, fungi also use this glycogen as the storage form of the sugar too. So that's the only difference. But here, the reason why starch and cellulose was so enlarged to show to you is just in case you wondered how come these two sugars, both of them are made of the same type of monomer, but they have such a quite distinct chemical property. One is like in our body, easily digested. The other one, cellulose, we don't have any enzyme to digest the cellulose. That's the reason why we cannot eat papers. We cannot eat grasses and then digest the cellulose, unlike some other animals like cows and giraffes and horses and sheep. They all can digest the cellulose, but unfortunately other animals do not. So the difference we can easily now then have a clue why. The reason is in that we don't have an enzyme to digest the cellulose, whereas those animals do have those enzymes. Yeah, that is the only difference. The cellulose, in terms of this intrinsic structure, it is very rigid. That's why the cellulose is utilized as a protective layer, like in the form of the cell wall, whereas a starch is a simply softer, flexible, but it can be easily digested, and also it can basically is a like a form of functioning, is a form of energy, this food source. But the main question is how come the same monomer, glucose, can generate such a two different type of structurally speaking only, like two different types of molecule. To illustrate, probably you can see even over here in this diagram if you are like take a look at closely, but I don't really like this looking at the details of the chemical structure. I just lied. I do personally love chemical structure, looking at the chemical structure for some reason, I don't know. But anyway, you don't really have to. So let me give you another kind of a way of distinguishing the difference between cellulose and starch. Let's take a look at this starch structure first. In starch, all individual glucose molecules are arranged in like having a chemical bond in this way, so to speak. Like if individual glucose molecule is something like is a human, then they have this hand-in-hand grab as a chemical the condensation reaction with a person next to you, hand in hand, happily just like that. It goes like so. All of the participating glucose monomers are looking at you, like in front of you, like they are facing the same direction, same side. As that's how all these glucose molecules are arranged in this starch molecule. Whereas in cellulose, one glucose molecule having this, hey, you don't look like smiling, smile, and the next glucose molecule is facing backside of, so showing his butt and back. And once again, the next glucose is facing the front, like alternating this side is what basically the cellulose in cellulose the monosaccharide, the glucose is arranged, and that type of different directions of this arrangement makes a huge difference in terms of digestibility. So we don't have any the native enzymes to be able to digest, and actually the fact of the matter is the only reason why cows and some other grass-eating herbivores can handle this cellulose digestion is only because not because they can make their own enzyme to digest, but simply because they can invite and then let them live in their guts the microbes, the bacteria and the microorganisms that can do this function for you. And also another actually surprising thing was a long time ago, human too, we were able to digest this cellulose because there is the evidence that a very long time ago in particular area of the colon, we used to have actually in our appendix. Usually in these days we just consider that appendix as an obsolete organ, so just consider that we should something that get rid of them sometimes. But actually inside of this appendix, there's evidence that those microorganisms was digesting the cellulose used to live. I don't know why we just kick them out. It's just a mystery. Otherwise, we would have been able to actually chew our the lecture notes while we are taking the lectures. It's fun, isn't it? And okay, here, this is another type of very similar polysaccharide, which is acting as a structural form. So it is not like the starch, but something more similar to cellulose. In other words, each component is also facing, each one is an alternating side. So one is facing this, for example, this sugar. If you are following the position of this oxygen on this sugar, this oxygen is up and this oxygen is down and up and down, like they are alternating like this. And we don't really have to perhaps go into this much detail, so don't really pay too much attention on what I am saying right now, especially if you are a non-science major. If you're a science major, actually, yeah, probably you better know, because you will have to go through this once again in the general biology anyway. So type of sugar is very much similar to glucose, except this part. This part contains, like, what difference do you see over here compared with the glucose? You have this nitrogen, it's never been in glucose, and also some of the particular it's called N-acetylglucosamine is named, but just because of having this a little extra part in terms of as compared with the glucose, that's why this sugar is called N-acetylglucosamine instead of glucose. But that's the only difference. Other than that, it is almost identical to the structure of the cellulose. So we can actually expect that this one could be used as another kind of a structural protection function. Yes, that is the case. But this one, so in particular, they are used in the outside protection of those insects. insects, unlike all these other the more familiar animals like humans and all other mammals and other spine-containing animals, like we call the vertebrate, they do have external skeleton called exoskeleton. So they have to molt. They have to get away with this their external skeleton at certain intervals. As they grow, their external skeleton will limit their growth, so every time they have to renew their outside this external exoskeleton, and that is exactly something that this I think it is a cycad undergoing this the molting, and also like shrimp and crabs, like those are the animals which is related with insect. The crustacean is named. They do also have the external this protection, like similar to this exoskeleton. Okay. So those materials are mainly made of this chitins, which is very similar to the cellulose. Okay, or with the exception of a slight differences in a certain part of the chemicals. That's all. And also, actually, the fungi. The big group fungi, the organism, they also have the cell wall, but their cell wall is not the cellulose, but this chitin is their cell wall material. So everybody else has cell wall, but the animals. Okay. Perhaps the final subject in the carbohydrate discussion would be the oligosaccharides. So some of you may actually think this oligosaccharides as one of the alternative form of the sugar tables, sugar, so that it's a healthier alternative of the sugar. That's actually what we use usually here. But the function in the cell in life is not at all about this related with this sucrose, anything like that. It is actually used as a very important marker, recognition on the cell surface. Very good example is actually a lot of times we practice this on many occasions. So when you go to a blind date, one of the most frequent topic is a subject of the conversation between your partner is what is the blood type, type A and type B, type O, AB. What's on the basis of what? This all type of oligosaccharide on the surface of your red blood cell. So depending on specific type of this oligosaccharide. By the way, the oligosaccharide means the reason why it is not called the polysaccharide is because the number of monosaccharide here is not something the range of several hundred or thousands, unlike the case of a polysaccharide. But it's not a disaccharide because it is not only made of two monosaccharide. So it ranges the number of carbons, the monosaccharides, consisting of these oligosaccharides is something like ranges several, 15 to usually 25, in some cases less than 10. I don't know. Something like a 10 to 25 is a more usual norm. Excuse me. So those are not too many and not too little number of those sugars having. So we just group them separately and then name them as oligosaccharide. Okay. And surprisingly, why do we have so many different types of oligosaccharides? It's because each different type of such oligosaccharide can distinguish itself, because structurally it will be all different. So it can be used as a very important recognition, especially if you put these different types of oligosaccharide uniquely, depending on who you are. Like red blood cell having this different type of oligosaccharide, make and then put on the surface of your cell membrane. And if you are some other, like for example white blood cell, then you do have a different type of oligosaccharide, so that you can distinguish, identify yourself as who you are, so that other like neighboring cell, just by sensing this oligosaccharide, then they can have a really good information. In that way, they can be used as a recognition marker. And also, as we can easily understand in the case of this blood type, blood transfusion, the reaction compatibility case, so our immune response will be based upon that, determining who is yourself and who is others, the foreign source, or mainly depends on this type of oligosaccharide. If those oligosaccharides are something that you already know, okay, then you consider that cell, the carrying that type of oligosaccharide as yourself. Okay, you're one of you. But otherwise, and then you just consider them as something to be destroyed. So that's how you use this oligosaccharide. So many different types of. So all these oligosaccharides, mostly of course, are attached to these proteins. These are two different types of proteins are shown on this figure. Some is just a simple, probably the receptor protein over here, and this one is a transporter protein. So in between, something is passing through like that. But not only that, some of the oligosaccharides are attached to directly on the membrane lipid. Membranes are made of basically is a lipid, directly onto some of the area of the lipids. So in there we distinguish usually the names of those glyco, the the oligosaccharide. The oligosaccharides that are attached to the proteins, then all together you call them as a glyco, which is a carbohydrate, glycoproteins. So the proteins having additional carbohydrates are names indicating the glycoproteins. On the other hand, glycolipid, which means a lipid having some of the oligosaccharide on top. So that's the difference between glycolipid versus glycoproteins. All of them are surface. All of them are something that having the carbohydrate, the oligosaccharide on their surface. Okay. So with that, I guess probably I should stop. I once again I made this all silly mistake of running too lengthy lecture. So it's been already way beyond 1 hour length of the lecture that I have originally intended. So I have to stop over here instead of moving on to the next topic, which is a lipid. So I will stop for a moment over here, and then we will see you in the next section of the lecture.